Hello everyone, my name is Rajneesh Mishra and I am a first year MBBS student. In this module, we'll be talking about carbohydrates, more specifically carbohydrates of physiological significance. So the 10 topics which we'll be discussing here are biomedical importance of carbohydrates, what are carbohydrates, classification of carbohydrates, glucose, isomerism in carbohydrates, physiological importance of monosaccharides, glycosides, deoxy sugars, amino sugars, and invert sugar. So let's start on these one by one. First, we'll be discussing about the biomedical importance. Okay, so we all know that carbohydrates are very significant in living organisms. What are the source of carbohydrates? Carbohydrates are produced in plants via photosynthesis. So carbohydrates are synthesized in plants by photosynthesis, right? In photosynthesis, what are used? CO2 and H2O. Second, in animals. In animals or humans, carbohydrates may be produced from amino acids. So this is not the significant way by which we gain carbohydrates. The carbohydrates in animals are predominantly the ones which are produced in plants, because we consume plant products. Which is the most important carbohydrate? Most important carbohydrate will be glucose, right? And glucose is the major metabolic fuel of mammals, except ruminants. Why? Because ruminants have adapted for other sources other than glucose, which include short chain fatty acids. Glucose is also a precursor for synthesis of other carbohydrates, including glycogen for storage, ribose and deoxyribose in nucleic acid, galactose for synthesis of lactose in milk, in glycolipids, and in combination with protein in glycoproteins, right? So glucose is precursor for synthesis of glycogen for storage, deoxyribose in nucleic acids, then galactose for synthesis of milk, ribose and deoxyribose in nucleic acid, galactose for synthesis of milk, in glycolipids, and in combination with proteins in glycoproteins, which is protein plus carbohydrate. So what are some diseases associated with carbohydrate metabolism? Diseases associated with carbohydrate metabolism are diabetes mellitus. Which hormone is involved in that? Right, it is insulin. Then galactosemia. Next one, glycogen storage disease. And the last one is lactose intolerance. right? Now we will be talking about the classification of carbohydrates, but before we classify carbohydrates, we need to understand what carbohydrates are. So carbohydrates are what? Carbohydrates are aldehyde or ketone derivatives of polyhydric alcohols. Aldehyde or ketone derivative of polyhydric alcohols. Now we shall begin with the classification. So first we can divide carbohydrates into monosaccharides, disaccharides, oligosaccharides and polysaccharides depending upon the number of constituting units, right? So we have monosaccharides, then we have disaccharides, then we have oligosaccharides and polysaccharides. Let's talk about these one by one. We shall start with monosaccharides. So what are monosaccharides? Monosaccharides are the sugars which cannot be further hydrolyzed. They are the simplest ones, right? Cannot be further hydrolyzed. Simpler carbohydrates. Then monosaccharides can be further divided into aldoses or ketoses depending upon the functional group. Okay, classified into aldoses or ketoses. Right, so let's talk about some common monosaccharides. First one are the trioses. They have a structural formula of C3H6O3. They will have 3 carbon, that's the important point. Aldoses with 3 carbon are glycerose or glyceraldehyde, and ketose is dihydroxyacetone, right? We have aldoses and we have ketoses. So the first aldoses are glyceraldehyde, or another name for the same is glycerose, and the ketose is dihydroxyacetone. After triose, we have tetroses. The formula is C4H8O4. They have 4 carbons. And aldose is erythrose, and ketose is erythrulose. Then we have the next one, that is pentose or pentoses. They will have 5 carbon, so this is C5H10O5. They will have 5 carbon atoms. Aldose is ribose, and ketose is ribulose. After pentoses, we have hexoses. they're quite important. Hexoses, pentoses and hexoses both are quite important. Hexoses will have 6 carbon, C6, H12, O6, right? 6 carbon atoms. So aldoses will be glucose, galactose and mannose, very common names. Aldoses are glucose, galactose and mannose, and ketose will be fructose. We'll talk about the relationship between glucose and fructose in the isomerism. For now, we can remember that glucose is an aldose and fructose is a ketose. Right. Next one is heptoses. Here we don't. They will have 7 carbon atoms, C7, H14, O7. Right, 7 carbon atoms. Here we don't have an aldose, so the ketose that is significant to us is ceroheptulose. Okay. Now one important point in this regard is in addition to aldehyde and ketones, the polyhydric alcohols in which aldehyde or ketone group has been reduced to alcohol group also occur naturally in foods, right? So the aldehyde or ketone group or the carbonyl group is reduced into alcohol group, and this occur naturally in food, right? I shall change the color now. Okay, so what are these called? These are called polyols or sugar alcohols. Sugar alcohols or polyols. They are synthesized by reduction of monosaccharides, and they are used for manufacture of food for diabetic patients, whereas food for weight loss, right? So how can they be synthesized? By reduction of monosaccharides. And where are they used? They are used as food for diabetic patient and also for weight loss. Important point is they have about half energy of sugar. Now next we'll talk about disaccharides. Disaccharides are condensation products of two monosaccharides, right? Condensation products. Examples of disaccharides are lactose, maltose, isomaltose, sucrose and trehalose. Lactose, maltose, isomaltose, sucrose and trehalose. Then we have oligosaccharides. Okay, now we're talking about the oligosaccharides. Oligosaccharides are condensation products, just like disaccharides, but they will have from 3 to 10 carbon atoms in them. Most of them are not digested by humans. Not digested by human enzyme, right? After oligosaccharides, we shall move to the polysaccharides. Polysaccharides are condensation product of more than 10 monosaccharides. Right? Examples are starches and dextrins. They may be linear or branched polymers, right? Linear or branched. Polysaccharides are sometimes classified as hexoses or pentoses depending upon the constituent monosaccharides. If they have hexose, then they will be called as hexoses, and if they have pentose, then they will be called as pentosins, right? Apart from starch and dextrins, there are also other polysaccharides which are called non-starch polysaccharides. And these non-starch polysaccharides are not digested by human enzymes and are major components of dietary fibers. They are not digested by human enzymes. Examples are cellulose from plant cell wall, inulin. So examples of non-starch polysaccharide: cellulose. Where is that found? In plant cell wall, right? Next example is inulin. That is a fructose polysaccharide, right? That is made up of fructose, and it is a storage carbohydrates in some plants.